We have a bit of an announcement before we start class today from your friendly representations from the Carleton Neuroscience Society.
Hi everybody, we are the CNS. We're a society just starting this year and a little bit last year, but we're trying to grow. We're trying to reach out to undergraduate students and hopefully get you guys involved and create more of a sense of community in neuroscience. However, we're not only just open to neuroscience students. We love to have cog psych, any psych students, anybody who's interested in neuroscience. And we are going to have our first event, a meet and greet at Oliver's from 6 to 9, or 6 to 8:30, sorry.
Yeah. There's going to be pizza and pop. Yeah, it's going to be free. Come for dinner. Yeah.
And I've heard you don't need to be of age. That's the whole point.
That's right. The point is that you guys can get in before.
Yes. So, if you have class till 5:30, good time to come and pick up some dinner and get to know more people in neuroscience if, you're interested in neuroscience and want to get involved.
If you just want to meet people in your major, because I know we tend to be pretty spread out and then we don't get to meet a lot of people.
Exactly.
That'd be pretty fun.
So, yeah, we hope to see you guys all there, if you guys can make it. Again, that's Ollie's on Tuesday, 6 to 8:30.
Cool. Thanks, guys. Yay, yay. How come you don't clap when I talk? You're welcome. All right, so good afternoon, everybody. It's Friday. Woohoo! So we're going to finish up today's lecture on neurophysiology, and I managed to get through quite a lot of content last class, which is awesome. And so hopefully that leaves us a bit of time today if you have any lingering questions or if you want to go over anything that we've talked about up until this point, if that needs clarification. So before we move along with going from postsynaptic potentials to action potentials, I want to make sure that we're all on the same page. So I have a little poll everywhere questionnaire. Ooh, fancy. So if you are able to pull out your cell phones and vote, for those of you at home, certainly if you're watching the live broadcast, you can absolutely vote in and see how your answer compares to the rest of the class. Certainly those of you that are watching it via VOD, you can do this and you can pause and get your cell phones out or log in on polleverywhere.com. So which of the following would increase the likelihood of cell firing? Would it be a change in the membrane potential from -70 to -72 millivolts? Would it be opening a chloride channel to allow chloride into the cell? Would it be opening a potassium channel to allow potassium outside of the cell? Or would it be a change in membrane potential from -70 to -65 millivolts? So for those of you that are not familiar with the technology, you pull out your cell phone if you are allowed to with your some service providers won't allow you to do that, and that's fine. You can log in. But you text the code to 37607, and whichever answer you think is correct, that's the code that you put into the message, and then send it, and we will compare answers. Yeah?
Something the potassium's going from in the cell to outside the cell?
It's allowing, yes, in to out. It's inside, we open a channel, it goes out. Is there Wi-Fi in here? I'm sure there is, hey? Yeah. So you guys can do it on your laptops if need be? All right, anybody still voting? Yeah? You can think about it, don't worry. Hold up your hand if you're still texting. Okay. Anybody still going? Okay. I'm just going to blank the screen here. Nice. Okay. Now, have a conversation. Find somebody next to you, around you, that gave a different answer than you and try to convince them that your answer is correct. And you're not to be talking about your weekend plans. Facebook is closed. Just kidding. Just a sec. Okay, now retext in your answer. If you've changed your answer, put it in as whatever you have changed it to. If you didn't change your answer, that's fine. Re-put it in.
Oh, actually, mine's on Sunday. Oh, yes, never mind. What? I'm good. I have no bar.
They're more confident in their answer. More confident. All right, anybody still going? It's not sending?
Just tell us what your answer is.
What do you think it is, Lee? Are you confident?
Nice. It worked. Yeah. So it is right. The most of you got it. But the first time around, it was only 60% of you, or 65. So excellent. So this is definitely why Poll Everywhere and this kind of peer-assisted learning works, because what happens is you explain to your peers in your own language why your answer is correct. And it's better than me telling you, because my language is on a little bit more nerdy side. So it's definitely something that I think is working. So awesome. Thank you for replying. Oh, we got some, 53 of you now. Anyway, so you guys online, I hope you, I know you don't have anybody to talk to about this unless you want to pause it and talk to your cat or your kids or your boyfriend, but you can certainly engage in the conversation by pressing pause and try to explain it in your own words, and hopefully it works for you as well. Okay, moving right along. So, so I just want to recap the summary that we had last class. That we were learning mostly about postsynaptic potentials. We first talked about the resting membrane potential, which is the fact that the inside of the cell is more negative relative to the outside of the cell. It's due to the imbalance in 4 different ions that we talked about. Oh, sorry, different ions. And there are 4 factors that contribute to this. This means that the neuron is polarized. It has charge. And this can be changed via these postsynaptic potentials, these small, local, graded, rapid changes in membrane potential, which is due to a change in ion distribution. And you can make it more polarized, so you can make it hyperpolarized, and these are inhibitory messages, or you can make it depolarized, which is excitatory messages, and these depolarizations increase the likelihood of cell firing. And then where I ended last class is I told you that if we manage to change the inside of the cell to at least minus 50 or so millivolts, so we make it much more positively charged, this is the threshold of excitation. And what happens is then that postsynaptic cell is going to fire an action potential. So what is the action potential? An action potential is a sudden reversal in the membrane potential from negative to positive, caused by a brief increase in the permeability of the membrane to sodium, followed immediately by a transient increase in the permeability of the membrane to potassium. On the right is a figure that is from another textbook that, of course, because of that, I cannot distribute it in your notes, but I'm going to take you through it because I think it's a really good diagram of what's happening at the action potential. So I think, yes. So here you see the threshold of excitation. Sorry, the resting membrane potential here is minus 70. If the threshold of excitation is reached, so if that entire cell membrane reaches minus 50 millivolts, what this does is it triggers voltage-gated sodium channels to open. So voltage-gated, meaning normally those sodium channels are held closed, but with a given change in volt, that's what sensitizes them, and they open their gate. Right? So they're voltage-gated sodium channels. What this means is sodium starts to enter the cell. Remember, sodium is more positive. It's a positively charged ion, so it's going to flood into the cell, and it's going to change the membrane potential to be much more positive. Right? So you can see here, if we're looking at membrane potential in millivolts, you see it starts to climb rapidly. Right? At some point, because the cell wants to return back to the resting membrane potential, it wants to go back to minus 70 millivolts, it's going to open potassium channels. Right? So those potassium channels are also voltage gated, meaning that they open at around, and this is like, I'm totally not sure exactly, but it's around minus 30 or 40 millivolts. Right? So it's when the charge gets even more close to positive that these potassium channels open, and as a result they start to allow potassium outside of the cell. So normally potassium is held in, those potassium channels, their gates, they open up, potassium starts to leave the cell because potassium is more positively charged. It's reverting the inside of the cell to be more negative. Okay, at around plus 40 millivolts, the sodium channels become refractory. And I'll explain what that means a little later on. But essentially what that means is they no longer are going to allow sodium out of the cell. They close, right? So they shut, and they're not opening anymore. But the potassium channels are still open. So potassium still starts to leave the cell. So now we see that we've reached the peak of the action potential, and we see the voltage start to revert back to its resting membrane potential. So potassium is leaving the cell, causing the membrane potential to return to resting levels. At some point, the potassium channels close, and the sodium channels reset. And we can see here that we have, it overshoots the resting membrane potential slightly, so it becomes slightly even more hyperpolarized. Right? So it goes below the resting membrane potential. And then the extra potassium outside the cell diffuses away. Okay? So the key is that the action potential is triggered at the axon hillock, that point where the cell body meets the axon. The axon hillock is rich in sodium channels, those voltage-gated sodium channels. So if that region of the cell reaches minus 50 millivolts, that's where all those sodium channels are going to open, and the action potential starts. And once it starts, it will travel all the way down to the end of the axon to the terminal buttons. And what you'll learn next week and the week after is where the magic really happens is at those terminal buttons, because once the action potential reaches the end of the terminal buttons, that's what's going to trigger the release of neurotransmitters. Rachel? Where's Anna? She's not here today. It's Na+. Sometimes I accidentally put 2 and she corrected me. It's actually just Na+. I was wrong. So it hasn't lost 2 electrons. It's only lost 1. Any other questions? So we're going to take you through all the different properties of the action potential, what it means. That's going to be the rest of this class. However, here's a fun question for you, and I know I'm throwing pull everywhere out because I think it's a handy tool. But here's a tricky question, very tricky question, and you're going to have to look at your notes to try and get the answer. The overall amplitude of the action potential is it 70 millivolts, minus 70 millivolts, 100 millivolts, or 1 millivolt? The overall amplitude of the action potential is what? Don't be freaked out that you're like, I don't know what the heck she's talking about. Just guess. And don't talk to your friends yet. Don't talk to your friends yet. Try to figure it out on your own first. It's not working? Do you want to use mine? That's fine. Oop. It's going to Facebook.
I'm liking the Dora the Explorer Band-Aid.
Yeah, I know.
Oh, that was my daughter. Thanks. All right. Do you want to put it in? Good eyes. Good eyes.
I have horrible eyes. I'm not
Oh,
wearing
really?
my glasses.
How do you know Dora the Explorer? Do you watch it?
I do. Every day of my life.
No shame. Are you sure?
Yeah.
It's going.
Slowly.
It just pauses.
Yeah, the signal here is really crappy. Oh, it's gone. It's gone through. Anybody still going? I see there's fewer here that are responding. You're a little unsure. You're like, Kim's throwing some wacky questions my way, and I just want to see if I put the right answer here. This isn't. All right, anyone still going? Raise your hands. You're trying? It's going. Ooh, interesting. Very interesting. No, it's where I expected you to be. Let's put it that way. Okay, so okay, now discuss. Find somebody that had a different answer than you. Okay, repoll. Did yours work?
Yeah.
Yeah.
And have to do it again.
It was slow, but this one worked.
My answer stayed the same.
But no, it's good to stay the same. Put it in. You can't go online? Go online. Yeah. Can you just Google it? You're not going to find the answer. Ah. What is the amplitude?
So it should be from the peak to the top, right? Not from the midpoint.
Okay, yeah.
You clever chickens. We still got 4 of you holding out here. Oh, 3 of you. Anyone still going? 2. Ha, interesting, interesting. No, it moved. So the correct answer is 100 millivolts. But in the first, when you guys first did it, it was almost like a 50-50 split. So at least it did move in the right direction. So just to explain to you why it is 100 millivolts, you need your figure here. If you look at the amplitude, so from basically what we're doing is looking from minus 70 or whatever it is here, but it's more minus 70 to positive 40, right? So that's about 100 millivolts amplitude. So from here to the peak, right? So 40 plus 70 is about 100, 110. Everybody understand that? Right? So it is definitely the case that novices will say it's 70 millivolts because they seem to get stuck on the 70 here. But once you understand what the amplitude is, then it's makes sense. So everybody get that? Or anybody else need more explanation? Yeah.
If it was like a short answer, long answer question, and we said 34 or 40, is that okay? Because I've been seeing like 30s in places and 40s in places.
Oh, for the peak?
Yeah.
Yes, absolutely. Like it's definitely not something, even resting membrane potential is never exactly minus 70. It's around minus 65 to minus 75. And the peak is never exactly plus 40, right? So I would never ding you on that, for sure. Oh, people are changing their answer. All right. So how do we get to an action potential? Keep in mind, most neurons receive hundreds of synaptic contacts, right? It's never just one presynaptic signal converging onto one postsynaptic cell. So to get to that threshold of excitation, what each individual neuron doing is it's summing. It's summing all the inputs into that cell and making the judgment based on the numerics of it as to whether it's going to reach that magic threshold of excitation. So we say what the neuron does is it integrates all the postsynaptic potentials. Remember, right at the beginning of term, I talked about the three main regions of the neuron: the dendrites, which are what receives the signal, the cell body, which is what integrates the signal, and the axon, which is what sends the signal. And that's what the cell body is doing, is it's integrating all these postsynaptic potentials. So individual postsynaptic potentials have very little effect on the firing of the postsynaptic neuron. And the firing instead depends on the integration of all the inhibitory and excitatory signals reaching the axon hillock. And as I mentioned, why it's at the axon hillock is because that region of the neuron is very rich in these voltage-gated ion channels. Okay, so remember the properties of the postsynaptic potentials. One of them is that they are graded. So again, all the inputs that are coming into the neuron, the closer they are to the axon hillock, especially if they're excitatory, they're going to have a greater influence on that cell firing. If they're inhibitory, they're going to have a greater influence on the cell firing, but in the opposite direction, right? So there's two ways that the neurons integrate signals. One of them is over space, and the other is over time. So we say that the integration is spatial or temporal. And remember, it's the influx and efflux of the ions is what's being summed, right? Keep in mind that the change on that voltmeter is not due to some magical property of the membrane. It's because of those change in concentration of the ions, right? So here you can see this is a figure that is reproduced from your textbook that show essentially the process of summation. So here in green are all the excitatory postsynaptic potentials that are converging onto the cell. And in red are all the inhibitory postsynaptic potentials here. So all these are summed by the neuron, reach the axon hillock, it depolarizes it to the threshold level, and generates that action potential. Yeah?
So each of the inputs isn't weighted like individually or wholly, like some are graded differently depending on how close they are to the axon hillock?
If we're talking about a spatial summation, yes, some will be more influential because the strength of the signal will be, it'll be a stronger signal because it doesn't have to travel very far. So remember, as the wave, the voltage declines as a function of distance. So any of the inputs that are way above on the dendritic tree are not going to have as much of an impact. So the closer it is to the axon hillock, the more of an impact it will have. And so that is spatial summation. So essentially, it's summing potentials that come from different parts of the cell. It is influenced by distance because the closer it is to the axon hillock, the more likely it will generate an action potential if it's a depolarizing current. If the overall sum of all the excitatory and inhibitory postsynaptic potentials can depolarize the cell, then this leads to an action potential. So here you can see these are the combinations of spatial summation. Here are two excitatory synapses, A and B, and here are two inhibitory synapses, C and D. Two simultaneous excitatory postsynaptic potentials will sum to produce a greater excitatory postsynaptic potential. So it's summing again over space because if this input is creating more positive charge here as well as here, then you're going to get a greater change on your voltage, right? Two simultaneous inhibitory postsynaptic potentials will sum to produce a greater inhibitory postsynaptic potential. And then if we stimulate all these at the same time, they'll cancel each other out. Okay, so all that is to say, the more excitatory postsynaptic potentials there are, all these postsynaptic potentials are going to be summed together to create an even greater change on the membrane potential. Questions? Confusions? People are looking at me very shiftily. What I see are all your eyes. You don't see anybody else's eyes. I do. And they're all like... Gabriella.
I didn't quite understand the postsynaptic part. So that's the postsynapse, which attaches to the axon hilum?
So these are presynaptic inputs. This is the postsynaptic cell. Any of the presynaptic inputs are going to have a change on the postsynaptic membrane. So that's why they're called postsynaptic potentials, because what you're measuring is the potential is the change in that cell membrane. So yes, it's influenced by the presynaptic input, but it's called the postsynaptic potential because it's that change in this cell.
Thank you.
You're welcome. So this is, the previous figure was not from your textbook. It's from a different one, but here's the one from your textbook for those of you that require or like having other visual information. Here you can see it's pretty much saying the same thing. We have input S1 and input S2. Two EPSPs produced at the same time but on separate parts of the membrane do not necessarily influence each other. So you can see they're located farther away from each other in space. Whereas if they're produced at the same time and close together spatially, they add to form a larger EPSP. Okay? So the closer the inputs are, which is kind of contrary to this one here, although they are somewhat similar. If it was B and D, they wouldn't have as much of a summation effect. Okay? So these are somewhat located further apart. That's why I prefer this figure to show exactly what I mean by spatial summation. Okay, so the closer the inputs are, remember, we're changing ion flow, right? So if they're located in the same general zone on the membrane, all those ions are going to have a greater impact because you're having a greater change in a greater portion of the membrane. So temporal summation is really measuring or summating inputs that come together close in time. So say we have one cell presynaptic input that fires and it changes the local membrane potential, and then 30 seconds later another one comes in, changes the local membrane potential. That's not going to have a great summating effect, right? But if we have one input, it fires right away after that, fires again, fires again, fires again, you're going to get this buildup of ions that is going to have a greater impact as to whether the cell will fire. So we sum the potentials that arrive at the axon hillock. The closer in time they arrive, the greater the summation and the possibility of an action potential. Importantly, you cannot have an EPSP and an IPSP from the same presynaptic input, right? Because it's at a single synapse. Everybody get that? So we have lots of inputs that are coming in, right? And with spatial summation, we're summing all these different inputs in space. With temporal summation, what we're looking at is one input over time. And you can't have a summed EPSP and IPSP with the same presynaptic input because that input is either going to release an excitatory signal or an inhibitory signal. It will never yield both. So one presynaptic cell firing onto the postsynaptic cell, it's either going to be inhibitory or excitatory. It's not going to be both. Okay? So here, imagine in this case, excitatory synapse A. If it fires in rapid succession, it will produce a larger change in voltage, right? So you can see here, A fires, and then soon after in time, it fires again. This one is showing a longer period of time. It's not going to sum as much. But if A fires, then shortly thereafter, it's going to, you can see it changes to minus 65 millivolts here. As opposed to here, it's only about minus 68. Same thing with inhibitory postsynaptic potentials. If they fire a long distance in time between one another, it's not going to have much change, but shortly after it fires again, you're going to have a summation on the inhibitory postsynaptic potential. I don't know who to take first. Jen.
So are you kind of saying that each terminal button, it will only ever be able to produce either inhibitory or like
excitatory signal?
Yeah, switch.
Yes, that's right. So for the purposes of this class, yes, let's assume that. Sometimes you can have what's called co-localization, but it's a bit of a complex thing that you'll learn probably in third year. But just know that for now, we're just talking about one input, and it can either be inhibitory or excitatory. So it's either going to release dopamine, and it's going to bind to a specific kind of cell receptor, or not. Other question?
So, sorry. Temporal summation, it's just, it has to do with only one signal, like not how far apart like two different like excitatory signals would be from each other.
Right, so
like
spatial
two
summation is summing all of the inputs over space, right? And what it says is that any two inputs that are close together spatially, and they're both excitatory, if they fire around the same time, that's going to have a greater change on membrane potential than, say, it's an excitatory input over here and an excitatory input over here. They're not going to sum. Temporal summation, one input, it fires, fires again, fires again, fires again, greater change. Fires, fires.
No matter what's firing around it. So
Right, but each neuron is doing both temporal and spatial summation, right? Yeah. So it is.
So like spatial kind of encompasses temporal with like.
Yes.
Yeah. It would. Yeah.
Yeah. So you're doing both. Yeah. Yeah.
I just understand like the big picture. So when you're like, see the ice cream or whatever, and you've got like some signals saying like I want to eat it, other ones saying like no, I'm on a diet. That's completely like separate. Like some of them are firing excitatory, some inhibitory. It's
Right.
Not like each cell is doing its own thing. Like will only do one.
Right.
So you have some that are go for it and some,
Right.
but you never have one
Like
neuron that will say both.
Here's the I'm on a diet. Eat me. Eat. 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 Right. And then this one might be your visual input. Then there might be another one, your olfactory input. One might be the thought of how much you weighed this morning. The one. So each of these is going to, like, and I'm making it extremely simple, but for the purposes of understanding this, sometimes taking it down to that can be helpful. But every input's going to be telling that neuron to do something different, right?
That neuron will only tell one thing.
That's right.
That neuron.
That neuron is going to be your motor neuron that's approaching, either approaching or moving away, right? So the key is if the sum of all the inhibitory inputs are stronger than the excitatory inputs, you will not go towards that ice cream. If the sum of all the excitatory inputs are stronger than the inhibitory inputs, you will eat that ice cream.
Okay, but then we might have more than one of these neurons though. So it's like we have some that like
Yes.
go for it and some that
More than one motor?
like yeah, exactly, yeah. Or would we only have one neuron that would
Yes, yeah. Yes, and that's why taking it very simply has its challenges. But yes, we do have populations of neurons that do these things. But keep in mind that all these populations of neurons are also going to have inputs from the same places. Gabrielle.
Can you give us an idea in terms of units of time? What does close, median, and far in terms of a temporal distance means?
Like in the order of half a millisecond is going to be short. 1 second long. So here's the figure from your textbook. So here are two different, this is a wide temporal spacing in excitatory postsynaptic potentials. And here you can see this is the input. S1 would be stimulus 1. And you can see here's, it's having a very little change on the resting membrane potential. But if we have them firing closer together in time, then we have a greater change, right? So they're summing, and so that the amplitude here is higher than the amplitude here. Okay? Even better, simultaneous input, right? See how closer we're getting to the threshold of excitation. Same thing with IPSPs. Yeah.
What if the presynaptic neurons, there are two of them, and they're far from each other, but they're firing really quickly? Does that have any effect?
So if the spatial information is far, but the temporal information is close, it's still gonna, you could still have a great impact, but it would be even better if they were close together and firing. Do you know what I mean?
Yeah, if it was the other way around.
Yeah.
Okay.
So you're getting at it for sure. High temporal summation is going to be good. High spatial summation is going to be good. High temporal and high spatial, even better. Oh, I have another one. I'm going to save this for later. Okay. So importantly, action potentials. So where we've gotten so far is we're integrating all these postsynaptic potentials at the level of the axon hillock. We're integrating over space and over time. Once these voltage-gated sodium channels open at the level of the axon hillock, then the action potential is propagated all the way down to the end of the axon. Now, students tend to think the action potential is a physical thing. They imagine it in their minds, and they see like a volt changing along the length of the action potential. No. What I mean by the action potential continuing all the way down to the end of the axon is that at every space along the axon, if you put your recording electrode into that membrane, you'd read out the amplitude of about plus 40 millivolts. Because what's happening is that all the way along the axon are sodium channels that are voltage gated. So what happens is that right at the axon hillock, you allow all these sodium channels, sodium into the cell. That's going to trigger the next sodium channels that are adjacent to it to open. So you allow all the sodium in, it makes the inside of the cell more positive. That triggers the adjacent sodium channels, and so on and so forth. So what I mean by the action potential traveling all the way down the length of the axon is that those sodium ions continue to trigger adjacent sodium channels to open, meaning more sodium is coming in and flowing in and flowing in and flowing in and flowing in all the way down to the end of the axon. Okay? So if you put in your recording electrode, here's the action potential, and you're recording down towards the direction of the axon terminals. This is what you'd see on your oscilloscope, right? You'd always have that change in voltage to around plus 40. By contrast, if all you had was a postsynaptic potential, something like a weak depolarizing stimulus, and you could do this artificially, so you deliver a very small change in membrane potential here, the size of the signal would decay to the end of the axon terminals, right? So you can see the signal is getting smaller and smaller. Okay? So that's why the action potential follows the all or none law. Once it is triggered at the axon hillock, it falls all the way down to the end of the axon. It's all or none. Once it starts, it flows all the way down to the end. Rachel?
In nature, are there ever any signals that get sent down the axon that aren't action potentials?
It can occur because, remember, we have axo-axonic synapses. So it's possible to have a presynaptic input onto the axon.
They would be flowing in the opposite direction, back to the hillock?
You'll learn that in third year. So what she's asking is, can it flow in the other direction? So that's called an antidromic signal, and it doesn't necessarily happen, but the complexity of that is a little above this level. So typically signals travel in an ortho. If anybody knows their Latin, ortho means right so in the right direction. So the signals go all the way down to the end of the axon. And what you'll learn is that it doesn't go back necessarily because of something special that we'll learn shortly. So the action potential always remains the same size without growing or diminishing. How is that different from postsynaptic potentials? What do postsynaptic potentials do? They get smaller. Why? What's that property called? They are graded. Beautiful. So on an exam situation, if I asked you, tell me the differences between action potentials and postsynaptic potentials, one of the things you might say is a postsynaptic potential is graded. The action potential follows an all or none law. Make sense? Don't give me your shifty eyes. Don't give them to me. So, in addition, action potentials are conducted much more slowly than postsynaptic potentials. Remember I said postsynaptic potentials are rapid? Because those signals reaching the ion channels, they open and then they're very quickly closed. But because we're allowing a lot of sodium in and we have to open the potassium channels and blah-de-blah-de-blah, they are conducted rather slowly relative to postsynaptic potentials. So another important feature of action potentials is they follow the rate law. And what that means is that because the action potential is all or none, and the size of the signal doesn't decay as a function of distance, variable information is represented by the axon's rate of firing. Okay, so the strength of the signal, the amplitude does not change. But what we can change is how quickly that neuron fires. Right? So if we have a weak input into the presynaptic input is weak, this is, if you can imagine, every vertical tick is a postsynaptic cell firing or releasing that action potential. So each of these represents an action potential. However, a strong input, okay, so imagine like bright light versus dim light, you're going to change that rate of firing such that that postsynaptic cell is going to produce an action potential at a greater rate. Right? So a high rate of firing causes strong muscular contraction, and a strong stimulus such as bright light causes a high rate of firing in the axons that serve the eyes. So that's how we represent variable information in the action potential. So it's not the amplitude, but the number of times a neuron fires for a given time period. Now keep in mind, of course, this is going to be influenced by presynaptic input, right? Or sensory information. The strength of the sensory information is what's going, or the strong stimulus is going to influence the cell. All right, a little bit now on refractory periods. Does anybody know what a refractory period is? Dave?
Basically like a period of time where the action potential can't occur because it needs like time to recuperate itself in a way.
Yep. So imagine, okay, you go flush your toilet, right? Flush. What happens if you want to do the double flush? You can't, right? There's a certain amount of time that has to happen before the tank refills with water, right? So that period of time between when you initially flush the toilet and then when you can reflush it is that period of, it's refractory. Another example is, toilet humor. It's not toilet humor, but maybe it is. Men's ejaculation, right? So men can ejaculate, and then there's a certain amount of time before they're able to have another erection, right? And so that refractory period in males, when you're 18, it's like 5 seconds. When you're 85, it's like a year. Okay, so it changes. And it's Viagra time. So this means that the toilet or the organism, it needs time to reset its system, right? So it's either water or ions, right? And that's what the neurons are doing. They're resetting those ion channels, and as a result, there's an amount of time right after an action potential fires, that the neuron cannot fire. And this is the refractory period. There's two of them. The absolute refractory period, and that occurs around 1 to 2 milliseconds after the action potential. And another action potential cannot be generated, right? So this is an absolute refractory period. And you can see this on this graph here. So here's the threshold of firing. During this time when the sodium channels are all open, potassium channels have just opened, this is the absolute refractory period, right? And then here during this time as well, once the sodium channels are starting to reset, then it's still absolutely refractory, right? So in terms of time, it's about 1 to 2 milliseconds. So another one cannot be generated no matter how strong the input. This is followed by the relative refractory period. You can see here, during this point when we've overshot the threshold of the resting membrane potential. And at this point, the action potential can only be elicited by very high levels of stimulation. So this is the way that the neuron codes for strong signals as well, right? Because if we have a really good, strong input into that cell, it's going to be firing during that relative refractory period. And so you're going to get that strong muscular contraction, let's say, or a strong blink, or withdrawal of your hand from a hot plate, okay? So we have the absolute and the relative refractory period. Incidentally, in males with erections, they have these as well, more or less. They have the absolute and they have the relative, but the relative is if it's a novel stimulus. So if they've just had intercourse with a female and another new woman walks in, that refractory period is going to be shortened. And that's called the Coolidge effect. And you'll learn a lot about that on the chapter on sex. But it is quite a wonder of science. That, and I think it was Dave. Was it you? Or maybe it was Jason?
It was me that brought that up earlier.
Yes, that said that, is this why men cheat? Was it you? Yeah.
Well, I brought it up in class at one point. I do remember that.
It was a
Yes.
girl.
I think it was Jason, 1201.
No, it was a girl, and then he disputed it. He said, "Oh, don't let her get away with it." It was the girl that brought it up first.
Oh, really? Oh, yeah, it was a girl. Yeah. Right. Comes up in every class. What can I say? So another important feature of action potentials, or something that can influence the conduction of action potentials, is whether or not that neuron is myelinated. Remember, the myelination is the myelin sheath that is formed by those glial cells. So what are they called in the central nervous system? Schwann cells? Who says it's Schwann cells? No one wants to guess. They haven't studied yet. What are those oligodendrocytes? Oligodendrocytes. The way I think about it is oligodendrocytes has the letter C in it. So does Schwann. Oh, I know how I remembered. I would say oligodendrocytes, and I'd say, oh, let's go to the central nervous system. So oligodendrocytes, they myelinate the cells of the central nervous system. The Schwann cells myelinate the cells of the peripheral nervous system. So a little bit about conduction in unmyelinated neurons. Like I mentioned, once an action potential has been generated, it travels all the way down to the end of the axon terminals, and they're triggering adjacent action sodium channels. So you can see here, this is kind of what I was explaining earlier. So we've stimulated, this is an artificial situation, right? So we've stimulated the axon here. You're opening those sodium channels, which are then followed by the opening of those potassium channels. And you can see those sodium channels are being opened all the way down the length of the axon here, right? So the size does not diminish as a function of distance. So that's in unmyelinated axons. So in response to every change in membrane polarization, this triggers the opening of the adjacent sodium channels, and another full-blown action potential is generated. Okay, so it's like waves of action potentials traveling down the axon. It's not one big action potential. The action potential is that change from very, very negative to very positive, and this occurs all the way down the length of the axon. Okay, so the refractory periods create a single discrete impulse that can travel only in one direction. So this is what Rachel was asking. This is why it can only travel in an orthodromic direction, because those sodium channels are refractory, right? So it's only opening the ones that haven't yet been open, because it can't travel this way because these sodium channels are refractory. Everybody get that? So you can only travel in one direction. Just checking for the shifty eyes. So this is why this is a relatively slow way to travel down the axon, right? Because we're waiting for all these sodium, these individual sodium channels to be opening all the way down the length of the axon. By contrast, when we myelinate the axon, it changes the conduction. So remember, this is what it looks like. The oligodendroglia. Remember, one oligodendrocyte myelinates multiple neurons. So you can see that in that figure here. By contrast, one Schwann cell myelinates one axon. So you can see this axon has multiple individual Schwann cells. So you can see each of this is the nucleus, right? So this is one Schwann cell, one Schwann cell. So we have lots of little Schwann cells. By contrast, one oligodendroglia has multiple outputs and myelinates lots of different axons. Okay? So this configuration means that we have a different kind of conduction. It's called saltatory. For those of you that know French or Latin, to sauter en français is to jump. So saltatory conduction is we're jumping from node to node. And what I mean from node to node is each of these individual, the node is the place in between the beads of myelination, right? So the myelination is not just one layer covering the axon, it's little chunks, right? And the spaces in between the myelination beads are the nodes. So right here, and right here, and right here. Okay? So here you can see this is the section of myelin. They're actually called nodes of Ranvier. So the action potential literally jumps from node to node. So we don't have an action potential here. We have an action potential here and here. Okay? And this is because myelination resists the flow of potassium ions. It resists the flow of potassium ions, so potassium ions can't leave that section of axon. So as a result, we're only triggering these sodium channels on adjacent nodes. Okay? So you can see how fast the action potential travels is a function of the myelin. It's faster, it requires less energy, and as a result, larger diameter axons and more myelinated axons will conduct the action potential quicker. So you can imagine at a functional level, places where we require fast transmission of information is going to be likely myelinated, right? So our optic nerve, where we're taking in information from the visual world, is going to be myelinated. Most of the axons that feed our muscles are going to be myelinated, right? Because we don't want to have that information travel slowly before we need to kick a ball or remove our hand from a hot plate. So places that require lots of myelination or fast conduction of information are going to be myelinated. All that is to say, most axons in the brain and most axons in the periphery are myelinated. Yeah? All right, so unmyelinated neurons, you should be able to describe how conduction occurs in those and differentiate that from myelinated neurons. So the last thing I want to... Oh, yay, we're done like super early. The last thing I want to talk about is just kind of to summarize, because again, students get very, very confused between the difference between action potentials and postsynaptic potentials. So I really hope by the end of today that is clear in your mind, and I hope you guys at home, it's clear to you. I know you can't ask me questions live, but I'm absolutely welcome to questions if you want to email them to me or come see me in my office hour. But you should really be able to differentiate between the two of them. So this is a figure from a different textbook, and I'm going to walk you through it. You should take notes because I can't give it to you, because I expect you to know this information, okay? So here's the different types of signals. Here's your action potential, and then your EPSP and IPSP. The signaling role, so what's the purpose of an action potential? Is to conduct information along the neuron. So it's really the main signaling property of the cell. So it's connecting it along the neuron, whereas the IPSPs and EPSPs are transmitting information between neurons. Okay, so from the presynaptic to the postsynaptic cell. You guys. It's all good. Is this allowed? Is this allowed? Yeah, it's fine. So typical duration in milliseconds, it's 1 to 2 milliseconds from the action potential, whereas the duration is typically longer in milliseconds for the EPSPs and IPSPs. Remember the amplitude here? Oh, looky, 100 millivolts, whereas we have a much smaller change in amplitude from the EPSPs and IPSPs, so it's along the order of 1 to 20 millivolts versus 1 to 15 for hyperpolarizing. We talked about this last class. The character of the action potential, it's all or none or digital, right? It either occurs or it does not. It's a one-to-one relationship between the signal and the output. Whereas the character of postsynaptic potentials are graded and analog, right? The strength of the signal is reflected in the strength of the amplitude of the change, right? The action potential is actively propagated down the axon, whereas these have local passive spread. So this is reflecting the character of those ion channels. In action potentials, we have first the sodium ion channels that open, then the potassium channels. Ion channels that open for EPSPs or IPSPs can be sodium or potassium. Potassium or chloride channels can also open for inhibitory postsynaptic potentials. It depends if this is an inward rectifying or outward rectifying current. And the channel is sensitive to voltage. Remember those voltage-gated sodium channels? Whereas these, what we'll learn next class, how these open is by virtue of chemical transmission or neurotransmitters. Okay? So I'm happy to leave this up for a while for those of you that are taking notes. I'm happy to answer any questions. And other than that, we will have, sadly, only one lecture next week because next Friday is CU Day. So we'll start in on neurotransmitters next week. We'll finish them up the week after that. And then the following Friday, and I'll remind you of this, will be the review lecture and then our midterm. So for those of you that want to leave, have a fantastic weekend. For those of you that want to stay and ask questions or take more notes, I'll be here.